In this video we will discuss about this HTPBD with this TRAP targeting plasmid enhance the chemotherapy that can cause this immunogenic cell death. So we will discuss how they come up to these conclusions. So first what they did is they use these HTPBDs with this TRAP1 plasmid into this cancer cells, that is CD26 mutated cells, and then they are able to see that with increasing concentrations the HTPBD with TRAP1 knockout plasmid or with TRAP1 targeting gene TRAP1 gene targeting plasmid has caused reductions in the cell viability compared to the control case, whereas for the normal cell lines, that is NIH 3T3, it does not cause any significant changes, indicating that the HTPBD can selectively target the tumor cells that has this HTPBDs can selectively target the tumor cells that has the mutations that makes them these CD26 cell lines, and by targeting this TRAP1 gene they can see a suppression or decrease in the cell viabilities, but not for the healthy cells. And these things they have validated using immunofluorescence also, where we can see that the HTPBDs alone can decrease the cell viabilities, but when it's combined with this 5FU, it can decrease the cell viabilities to much more extent. But when they use this CSA, they can see an increase in the cell viabilities. And these things they have quantified in the panel D, where we can see that when they use HTPBDs with TRAP1 targeting plasmid and 5FU, they can see a maximum reductions in the luminescence, indicating maximum reductions in the cells, compared to the case where CSA is used with 5FU, which shows reductions of around 35%. Then they wanted to check whether it is causing this cell death, immunogenic cell death, or this reduction in cell viabilities is because of the mitochondrial damage or not. So in order to check that, they use these JC1 assays, where JC1 aggregates indicates a healthy mitochondria and JC1 monomers indicates a damaged mitochondria. And we can see that when they use the control case, we can see a healthy mitochondria in the tumor cells, and when they use the 5FU only or the HTPBD vectors, the results are for the HTPBD vectors, the results are not different compared to the control, but for 5FU we see a slightly decrease in the aggregates and slightly increase in the monomers signals. And when they use HTPBD with this TRAP1 targeting plasmid, we can see increase in the monomers and a decrease in the JC1 aggregate, decrease in the mitochondria aggregates, but this decrease does not look like significantly different compared to the 5FU. But when they use HTPBD TRAP1 plus 5FU, they can see almost negligible signals for these JC1 aggregates and high intensity signals for the JC1 monomers. But when they use CSA, they can see suppressions in the effect of this HTPBD TRAP1 plus 5FU, and they can see some JC1 aggregates, healthy mitochondria, and decrease in the JC1 monomers. When they use the TEM image, they can see that for the control case they do not see a cell death, but for the HTPBD TRAP1 they can see some of the cell lysate, and similarly for this HTPBD TRAP1 plus 5FU they can see some of these cell lysates that are marked by the red arrows. Then they wanted to check this mitochondrial healthiness using different approach, that is using calcine approach. Calcine can calcine is kind of we can say that this calcine can enter first they introduced a non-fluorescence dye in the cells that can go in the cytosols and also in the mitochondria. Then they use this calcine that can also goes to the cytosols, but it cannot enter the healthy mitochondria. So this is how they distinguished the calcium signals between the healthy mitochondria and the damaged mitochondria. And when they did their studies, for the control we can see a healthy mitochondria. For the control we can see the signals for this signals for this calcines in the cytosols, that can be seen as around 96%, 93.6%, and for the control case they can see 99.9%. And when they use HTPBD vectors without without having the plasmid that target TRAP1 gene, they can see the signals of around 91.4%. But when they use 5FU, they can see reductions in the signals. Reductions in the signals in the cytosols. We can see that the reductions is around 43%. When they use HTPBD TRAP1, they can see reductions of around 46%. This is similar to the 5FU. But when they combine HTPBD TRAP1 and 5FU, the reductions goes down to 21.5%. Then when they use the CSA plus HTPBD, they can see some recovery. In the CSA plus HTPBD TRAP1 plus 5FU, they can see some recovery to around 39.3%. And for the positive control, they use inomycins, where the signals goes down to 16.4%. And then thereafter they also check the effect of this cell death on the immune activations. So they check the dendritic cells maturations from CD80 and one more markers, and this dendritic cell maturations is from the bone marrows, and they can see that the maximum maturations that they have observed when they use this HTPBD TRAP1 with 5FU. Results are highest for that, and some of the control results are shown here, where they use BIPV5 and BAL1 with HTPBD and 5FU. So this indicates that this HTPBD TRAP1 can be combined or synergetically used with other immunotherapies to increase the specificity of the treatment. In the penalty, let me again recall to you that calcium shows the signals in the cytosols. So once they are treated with this 5FU, then the calcium will go, the calciums that are in signals that are in cytosols might enters the mitochondria, and the cytosol signals goes down. This is how we come to know that the damaged mitochondria is present using this calcium based assay. I hope you understood how they check the HTPBD's effect. HTPBDs with TRAP1 targeting plasmid has a positive effect to other chemotherapies such as immunogenic cell death using 5FU, and that has been shown here. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.